Hello again, students. Welcome to the third and final installment of the Chapter 14 lecture. This slide shows basically how radiocarbon dating works, and the example here is radioactive decay of carbon-14, which is an isotope of carbon. So from the time an organism dies, it takes 5,600 years for half of the carbon-14 in its tissues to decay. This is what's known as the half-life. So carbon-14 has a half-life of 5,600 years. So once that 5,600 years has passed, half of that carbon-14 has decayed, and then half of the remainder carbon-14 within the tissues is present after another 5,600 years, etc. So this graph just shows that visually. It shows carbon-14 radioactivity on the y-axis as a percentage of the living organism's carbon-14 to carbon-12 ratio. So time is given in thousands of years on the x-axis here, and when you start at time 0, which would be right at the time of death of a certain organism, it's still going to have 100% of its carbon-14 radioactivity. As time goes on, you're going to have less and less carbon-14 in those remaining structures. So again, after 5,600 years, 5.6 here, in this graph, we have 50% of that carbon-14 radioactivity present. So it decays at a very predictable rate, and this is the basis for carbon dating methods. This is another representation of that process, and we have a shell here that's buried in sediments. Of course, that shell was once part of a living organism that had soft tissues, so it could be a clam. Carbon-14 is taken up by the clam in trace quantities, along with much larger quantities of carbon-12 during its lifetime. After the clam dies, carbon-14 amounts decline due to this radioactive nature. And then when people are discovering these preserved fossils, you can measure the ratio of carbon-14 to carbon-12, and that will reveal how many half-life reductions have occurred since the clam's death. And I would also like to note that these blue dots within that clamshell represent the amount of carbon-14 that's still present. So this is just one example of how radiometric dating can work. Of course, depending on the specimen and depending on how old it is, you can look at different isotopes besides just carbon-14. Here's a few really cool examples of fossils. If you've ever heard of petrified wood, that's also just known as fossilized wood. So you can find parts of trees that have been fossilized in some parts of this continent. And this particular example is a sedimentary fossil. It's formed by minerals replacing the organic matter of a tree. We also have trace fossils, which show oftentimes footprints, burrows, or other remnants of an ancient organism's behavior. We have the famous amber example. Of course, Jurassic Park-esque technology has not been developed, nor do I think it is actually possible to extract intact DNA within these species, but this particular example is an insect embedded in amber that's 45 million years old. Here we have a researcher excavating a fossilized dinosaur skeleton from sandstone. And here we have tusks of a 23,000-year-old mammoth discovered in Siberian ice. Now, these remains here are not actually fossilized, they are just very well preserved in ice. So oftentimes, especially with climate change, there will be discoveries of ancient mammals, and in many cases the soft-bodied structures such as hair and skin are actually remarkably well preserved just because they've been entombed in ice for thousands of years. Okay, so getting back to this macroevolution concept here, plate tectonics play a huge role in that. And plate tectonics are responsible for many of the modern-day patterns that we see in biogeography. Land masses are not static, and continents sit atop irregularly shaped plates of crust which float on top of a hot, flexible layer of the Earth known as the mantle. Currently, North America and Europe are drifting apart at a rate of about 2 centimeters a year, which is not a big distance, but when you consider that the Earth is 4.6 billion years old, plate tectonics can actually account for vast distances being formed between different land masses. And we also have other types of events besides just plates separating that are also responsible for evolutionary forces, such as violent upheaval when two plates collide. And in this case, mountain ranges can form, which also cause volcanic eruptions and earthquakes. The Cascade Mountains in the western United States, which is basically where I grew up, is a great example of one of these mountain ranges. And there are volcanoes, both active and inactive, that form the Cascade Mountain Range. This is a schematic showing what the present-day Earth looks like, and if we go back in time, we can trace the routes that these continents took as a result of plate tectonics. At one point in time, we had a supercontinent known as Pangaea, and all of the modern continents are there. They're just slightly different shapes, and they're all connected to one another. Pangaea formed about 251 million years ago, and due to the movement of tectonic plates, we eventually had Pangaea splitting into two land masses known as Laurasia and Gondwana. Gradually, these continental masses shifted due to plate tectonics, and this is the ultimate cause of the landmass position in the present day. The pattern of continental separations is the solution to many puzzles of biogeography. And a few examples of this are matching fossils of Mesozoic reptiles have been found in Ghana, which is a country in Africa, as well as Brazil. These continents are currently 3,000 kilometers apart, but were a single landmass during the time period that these fossils formed. So as you can see here, Africa and South America were once joined, which will account for some of the similarities in organisms we see in fossils today. And of course, marsupials in Australia, they are unique because Australia had separated from the rest of the land masses early on in the course of mammalian evolution. So it wasn't until the land masses were separated did mammals actually start to evolve. And that is one reason why Australia has a very unique complement of mammals. Something that's important to keep in mind is that we all think of mass extinctions as being really unfortunate events and responsible for wiping out large portions or large sections of life on Earth. But you can also think of mass extinctions as leading to new opportunities. There have been 5 mass extinctions that are documented in the fossil record, and current evidence points to the fact that we are actually in a 6th mass extinction that's largely human-caused. A mass extinction is an extinction event during which 50% or more of the species died out. During the Permian extinction, 96% of marine species went extinct, and during the Cretaceous extinction, most dinosaurs died out. The lineage of dinosaurs that led to birds, however, survived. So what do you think happens when a mass extinction wipes out so many species? Well, probably for a great deal of time after that mass extinction, there's just a vastly reduced biodiversity on Earth. But over the period of evolutionary time, remaining populations will reproduce and eventually lead to new species. So when such a large extinction occurs, there is going to be an opportunity for other species to become more prevalent or dominant on the landscape. So even though mammals existed for 75 plus million years prior to the extinction event that led to the dinosaurs being wiped out, mammals only diversified and evolved into larger species after the dinosaur extinction. So it's important to keep in mind that mammals did exist during the time of dinosaurs, but dinosaurs were really the dominant presence. They were occupying all of the ecological roles, and they were very successful doing that. There wasn't a lot of opportunity for mammals to diversify and become larger because those dinosaurs were occupying the ecological roles, and they were doing that really well. It wasn't until that mass extinction event occurred that led to new opportunities for speciation in early mammals. And this next slide does not pertain to this course. It was for my last course that I taught, so this is going to be the conclusion of the Chapter 14 lecture. I hope you enjoyed listening, and again, if you have any interest in evolution and learning more about how evolution works, I strongly suggest Googling Your Inner Fish. It's an incredibly well done documentary, and it's actually really entertaining too. And of course, you can also consult your book, which will give you more details about all of the processes that I've talked about today, and feel free to use that peer-to-peer discussion board on Blackboard as well. Thank you for listening.